Hello and welcome back to Paleocast. I'm Liz Martin Silverstone.
And I'm Dave Marshall.
And this is episode 116 on Ice Age Paleoecology.
So
Dave,
Liz,
what's new? Oh,
no, I'm saying hello to you. Liz, it's been a while. How have you been?
Yeah, it has been a while. I've been pretty good. Slowly surviving lockdown, I guess. Or not really lockdown, but pandemic. I got a new job, so that's quite exciting.
Fantastic. Tell us all.
So I am now the lab manager in the University of Bristol Paleobiology group. So I now get to actually work as a kind of proper paleontologist doing lab things, which is quite fun.
Awesome. And when you say lab things, what does that involve? Because it can be a hugely varied job, I guess. Some people are chopping up alligators and looking at the bones. Other people are sticking together bits of scanning electron microscopes. What does it mean for you?
So for me, I'm the lab manager, so I'm in charge of looking after all of the labs in the paleo group, which it includes the CT scanner is kind of the main thing. So I run and manage the CT scanner, and also all of the wet labs, so for dissections or any molecular paleobiology, kind of more traditional proper lab stuff, and also the micropaleo labs, so making sure that everybody's got what they need for foraminifera picking and sampling and all of that side. So I'm responsible for looking after a large number of labs, which is also slightly terrifying.
Nice.
Yeah.
But congratulations.
Thank
from
you.
me and from all of our listeners too.
Thank you. It's very exciting. It's something I wasn't expecting, so I'm still kind of getting used to it. But what's new with you?
Nothing. I've just been sat around doing not very much, and that has been fantastic. Pandemic for me has mostly involved me sitting around playing computer games for the Paleocast Gaming Network, which of course everyone is aware of and subscribed to on YouTube and or Facebook, and all social media. And then otherwise, like, what's turned out to be my full-time job pretty much now is our virtual natural history museum project, which those of you who've been hanging around for ages would be aware that this has been something that's been on the back burner for such a long time in Paleocast, and it's going to be a big computer game museum that is literally a computer game. You run a little character around and go and look at real fossil specimens. And so Tom Fletcher and I have both been pretty much doing that every single day. Every evening we just get on Discord, we sit down, we add new fossils to the museum, we run around, have a load of fun, and it's getting close. We're getting there, and it's gonna be months now until it's ready, and then it's gonna be ready, and it's gonna be fantastic. And you can run around the museum and go and look at, like half of the world's digitized fossil specimens. It's gonna be fantastic. So really excited about that.
Yeah, that's really exciting. I can imagine that while people are not spending a lot of time going to museums, this is actually kind of a good time to get this out and be able to show people some fossils.
Yeah, definitely. But it's a huge undertaking to, kind of write about the whole history of life on Earth. So yeah, if we've got any paleos listening, get in touch. We could do with some extra volunteers. But that's enough chatting about stuff that we're doing. What's the episode on today?
This episode is with Jacqueline Gill, who's an associate professor in paleoecology at the University of Maine. And she had just released a paper around the same time as when this episode will be launched. So we were talking a little bit about what her paper was and some of the work she's done on environmental changes and understanding interactions between plants and animals, and what this has to do with sort of local landscapes and different ecological interactions around kind of the Ice Age all the way up to now. So it's something that I think is really interesting to understand what sort of past ecological interactions can tell us about moving into the future. And one big takeaway that I got from this is how much poop can tell us about things.
Yeah, there was a lot of poop chat. And,
I had no idea how much you can learn from poop, so that was exciting.
I knew there was a lot, and many of the other paleo podcasts out there, it's one of the first things they go to. It's like, coprolite episode, this is fossil poop, and you can learn so much from it. And it's not something that we've actually ever touched upon here.
I feel like we could have I definitely feel like we could have a poop episode just in general in the future.
We totally could. We could have multiple, but there's still a lot of other content to talk about. And what really got me from this episode was the interactions between plants and animals, and how you would just by default think, say climate change affects the plants affect the animals, but it might actually be the other way around, that animals are the driving force behind some of the changes that we see in plants. So
yeah, it's true. You don't really think about it going both ways, and I think it's really interesting to hear all the different interactions that work. So I hope that you can learn something from this episode as we did.
And as always, we've got pictures on our website to accompany this episode, and if you can give us a little boost on social media, that's always appreciated. Shares, likes, comments, subscribes, what are the other things they can do? Save on Instagram, that's the big one.
Yeah. And
we'll issue 10 Paleocast points for each one.
So if you want to hear anything else from Jacqueline, she's also got her own podcast called Warm Regards. So if you're interested in more information about climate change, you can go and have a look at that. She's also quite active on Twitter, so have a look there, and we hope you enjoy this episode. All right, so today I'm sitting down with Dr. Jacqueline Gill, who's an associate professor from the University of Maine. Thank you very much for joining me today, Jacqueline. I'm really glad that you could come on the show.
I'm really excited to chat with you today.
Can you start out by kind of introducing yourself and telling us how you ended up in your position in Maine?
Yeah, I am a palaeoecologist, which means I study past ecosystem changes and species interactions, which we reconstruct mostly forensically. So unlike a lot of my colleagues who are paleontologists who work in the vertebrate record, I work mostly with sediment cores from lakes and bogs. So we use things that are mostly microfossils, so pollen, spores, geological or geochemical indicators in the sediments. And part of what drew me to this field was that the tools are so creative, because we can't just go out and study our ecosystems or organisms. We can't just observe them in person. So we have to use all these forensic tools, which have always been super fascinating to me. I just love all the indirect ways that we have to kind of piece together these past changes. Like it's very much like a puzzle to me. And I always I went through so many different possible career pathways. For a while, I went through my marine biology phase, which I think a lot of us go through. I wanted to do history, photojournalism, theater, communication. So eventually, but I always had this strong interest in the natural world, in conservation. And so when I, really started to kind of find my passion, it was very much for understanding, how the past can help inform our understanding of ecology and conservation biology today. And I was really fortunate I went to a small liberal arts school called College of the Atlantic, about 325 students, very interdisciplinary, very liberal arts. And that kind of set me on this pathway of kind of getting to do a little bit of everything, and really being in places that value cross-disciplinary collaborations, that value the humanities, that value the social sciences, value the arts. So from there, I went on to a geography department for my master's and PhD, which here in the US, geography is not as robust of a discipline as it is in the UK. A lot of our programs closed in the 80s, but I think it's seeing a resurgence because of all these interconnected global change problems. And so when this job opened up at UMaine, it was a split position between the Climate Change Institute and the School of Biology and Ecology. It was for paleoecologists working with sediment cores. It was in New England, which I love, and it was a perfect fit. And I was just really fortunate that the timing worked out, which just never happens in academia. I had been on this kind of interdisciplinary path for a really long time, and the timing just really worked out as it sometimes does.
Yeah, it sounds great. I mean, it sounds like you're quite lucky to get that at exactly the right time, which is awesome.
Yeah, it was really fortunate, because, yeah, I mean, I as paleoecologists, we can end up in lots of different places. I have colleagues in
Mm.
geology and ecology and environmental science, anthropology, geography, and I'm just really grateful to be in a place where, UMaine has a really long history of supporting interdisciplinary research, and the Climate Change Institute is 40 years old. We used to be a quaternary research institute, and then we kind of broadened out a bit because of concerns about modern climate change. And so, yeah, I'm
Mm.
just really glad to be in a place that that kind of has this long history of valuing work with people across departments and disciplines.
So, why paleoecology? What do you think is the most kind of exciting bit about that, since that's obviously what your research mostly entails?
Well, for me, it's kind of as I said, a little bit about the methods. It's just
Hmm.
these indirect indicators that we have to use to reconstruct past ecosystems and communities. The tools to me are creative and they're fun. I just, we were working with pollen grains and tiny flecks of charcoal and poop that grows on, or sorry, fungus that grows on herbivore poop, and it's just these really interesting tools that in and of themselves, I love the methods, the approach, the reconstructing timelines of change, is I think a big draw for me. The dynamics that the dynamic perspective that a long term study on ecosystems brings. Because when I was a student coming up, and learning about ecosystems and learning about ecology and conservation, I kept running into these really static perspectives on what
Mm.
communities should look like, right? That there's a climax community or a balance of nature, or that we should be aiming for a conservation target that was this, like here in the U.S., it's often pre-European contact, which sort of assumes this kind of
Mm.
empty landscape of a wilderness that was devoid of people or devoid of change. And but then as I was learning more about geologic time and even just the recent past, I was really struck, especially here in New England, about by just how much change had happened: sea level rise, glaciers retreating, different tree species migrating in and out, species going extinct, new ones arriving, and all of those things just clashed so much with what I was learning in my contemporary classes. And I
Mm.
felt like so much of what we think we know about the world is based on this really static view and this short-term thinking. And it seemed to me like a lot of our problems, whether we're talking about politics or species conservation, could really benefit from longer-term perspectives. And to me, the change in the dynamic, nature of these communities you know, is part of their strength and their resilience. And I wasn't hearing enough about that. Um,
Mm.
from my classes and from the policy work I was engaging in, and the activism I was doing as a student. And so for me, it was like, okay, this is where I want to go. I want to study these past ecosystems, not just because I think they're fascinating, and I still resent the fact that we came so close to living in a world with woolly mammoths, but we don't. I will never let go of that resentment. So they're interesting in and of their own right, but also they have so much to teach us about so many of the problems that we're facing today as a society.
Mm. Do you typically take all of these different methods and these different types of data that you go into the field and analyze? Do you do that for a single ecosystem so you get kind of a big picture, or are you looking at maybe fungus on a certain thing over many different ecosystems to see the differences between them?
Oh, that's a great question. We do a little bit of both.
Hmm.
We have some projects where we look at, we kind of stitch together lots of these paleo records across geographic space. So we come up with,
Mm-hmm.
what I like to think of as a 4-dimensional picture of species through time. So you have space and time, and, which talk about, ways to bend and break your brain, like try to imagine and visualize data in 4 dimensions. Yeah, so sometimes we might look at the migration of a forest species across space through time. Um,
Mm-hmm.
but other times, we will want to have a very multidimensional picture of a place where we try to put together as many possible components of that community as we can. And I started off working with lake sediment cores and the pollen record when I was in graduate school, and working with these dung fungi kind of added this extra element of herbivores to that story. And but we were very limited when I was just starting off by, okay, we can mostly understand plants. Combining plants and animals is super hard, and, let alone insects or anything else you might be interested in. And that has changed a lot just in the last decade in terms of some of these new and emerging tools, whether we're talking about ancient DNA or other kinds of proxies to reconstruct past ecosystems. But we're also starting to work in new kinds of systems, so cave deposits, or we're wrapping up a project at the La Brea tar pits where you have many, many different components of that community in the Los Angeles basin that are all preserved in these asphaltic sediments, or the permafrost tunnels that we work in Siberia, where you've got birds and moths and plants and animals, and if somebody wanted to look at their parasites, we've got fur, we've
Mm.
got gut contents, right? So there's just this explosion of possibilities now where I guess I like lots of dimensions. I like thinking
Mhm.
in a big picture way, whether that's, okay, let's understand this place from as many different angles as possible, or let's just look at the whole continent and see how species are migrating back and forth in response to climate change.
What are the advantages of using these kind of multi-dimensional studies over maybe other more traditional single data point methods?
I think both are extremely important and they really need each other, right? Both of these
Mm.
approaches. I am the sort of person that I cannot just focus on one place or one thing for a long time. We've got projects in the Falklands, Jamaica, Siberia, North America. We're all over the place. But but I have colleagues who spend a lot of time on a single organism or on a single
Mm.
fossil locality, and I think there's this nice trade-off in terms of what you can learn. We need really strong, good natural history observations and good species specific or site specific data to help us contextualize and understand the processes or the processes that drive the bigger scale patterns that we observe. So I sometimes people talk about how we don't even really need species, we can just think of them in terms of their traits, um,
Mm.
or or even their genes nowadays. But I think I think that there's valuable information there, but I think that I don't think we should give up one of these perspectives, right? I think we need dialogue among them all because we'll see
Mm.
we'll just be able to understand what we're looking at so much better, because the challenge that we have as paleoscientists is that we don't get to see our study systems in person. And so,
Mhm.
because we have to use these forensic tools, um, linking cause and effect or pattern and process is incredibly difficult. And the ways we get around that are by doing modern experiments when we can, which isn't always possible.
Mhm.
I try to do that when possible, but it's, you can't really do a lot with a trilobite, right? There's
Yeah.
not, maybe I can work with bison today, but that doesn't get me, as far as working with an elephant, right? Um,
Mhm.
so yeah, you can do modern experimentation. You can really precisely try to delineate the order of events. So did something happen before or after something else? So having high resolution multi-proxy studies, and that can help you weed out possible hypotheses, but it still doesn't get you quite as far as a modern observation might. And then just having more kinds of data so that you just have a stronger, deeper
Mhm.
understanding of the natural history of the species that you're working with. And so so I think the big, so I guess I haven't answered your question yet, partly because I wanted to say, like, my choice of scale that I work at is really driven by the questions I'm interested in, but they
Mm.
rely and just my brain and how my brain works. I mean, I listed off like 15 different things I wanted to be when I grew up, so it probably didn't give you any, like, there should be no surprise that I'm, interested in lots of different things. Um, but I think, the scale that I choose to work at is the one that makes sense to me, but
Mm.
I rely so much on, what some of the more traditional approaches in terms of, working with a single specimen or organism or species or locality. But I think when you zoom out, you can see some really neat big picture patterns,
Mm.
that are also not possible, when you look really closely at something. And so it's like the forest for the trees metaphor, right? Sometimes the questions that we're interested in really mean that we need to focus on the trees. And sometimes,
Mm.
but the questions that I tend to be interested in, which is what are the lessons we can learn from the fossil record for conservation? How
Mhm.
can we inform our modern ecological theory with, and vet that against these long time scales? I find that the answers for me for some of those questions about things like community assembly, why we have
Mhm.
so many species coexisting, what controls ranges at large spatial scales, a lot of those kinds of questions, those for me are more tractable at the larger scales.
That makes sense. And I guess, yeah, when you're looking at things that are kind of trying to understand how the past can give kind of pointers and help us in the future, it makes more sense than just looking at a single species, because that's not going to tell you a whole lot about what's going on in the next 1,000 years.
Yeah, or but I mean, it might help inform, conservation or our understanding of the long-term ecology of that particular taxon, if that's what you're working with.
Hmm.
Yeah, so I think it can be, like I said, I think we need more dialogue, and sometimes I see sometimes I see people arguing that one approach is better or worse, or I see people saying, the future is in data science and we don't need to do a
Hmm.
lot of this on the ground, work, or we don't need collections, or and yeah, I think that's really myopic. And then I see people, turn around and say nasty things about data scientists too, where, oh, you're
Sure.
swooping in and taking all of our hard labor and then, doing some statistics, and it's,
Hmm.
without appreciating all that goes into what we do. And I think that those kinds of arguments will, they go away when you actually have more friends and colleagues who do those kinds of, we all need to work together, right? So
For sure. I guess thinking to some of your research, you had mentioned before about never being able to see a woolly mammoth, and going into some of your work on the more kind of larger scale paleoecology questions. You've done quite a lot of work in the Pleistocene and sort of Ice Age paleoecology. Why is that time period interesting to you,
oh yeah.
other than wanting to see a woolly mammoth?
That's most of it. Yeah, it's one of those things where it's just funny, you just sort of find your place and then you click and you just know that's where you belong.
Mm.
And so for me, like I very much feel like I belong in the Pleistocene. And part of it was through, like, the hook for me was through these amazing large animals, because we've downsized so much as a planet now. These big animals in the past just hooked me, and then the more I learned about them, I mean, they're just so fascinating. Like here in North America, just the fact that we have beaver the size of we would have had beaver the size of black bears, and armadillos the size of a VW bug. I mean, that's just a really neat hook. And there's something
Mhm.
familiar about them since they're mammals. For me, I guess this is going to be sacrilegious, but I I think dinosaurs are interesting, but they never captured my imagination in quite the same way. And I don't know if it's just that mammals are more accessible somehow because we have them. So there's something that's familiar enough about these landscapes that makes you feel like you could be at home there, but they're different enough that they're fascinating, right? They're very much not home. And I like to tell people that if you were to travel to any point in time in the Pleistocene, you would feel like you were here, right? You might not even
Mm.
know something was weird until um and until like a woolly rhino lumbered by or something, right?
Mm.
That the difference really is one of omission, right? We are missing things. We are missing Pleistocene species. We've had very few new things crop up in this on this geologic timescale, and so we're talking about functionally modern ecosystems, and they're also ones in which we as a species emerged, and
Mhm.
we emerged from these Pleistocene ecosystems, and our civilization came out of these ecosystems. And when I talk about the sort of near miss of the opportunity of having those interactions with woolly mammoths, um, I like to remind my students, for example, that mammoths were around until just a few thousand years ago on Wrangel
Mm.
Island. These were, full-size woolly mammoths, and they would have coexisted, or they would have been around during the time of the pyramids. So when we had modern human technologies like writing, like the wheel, so we could, in theory, have had mammoth-drawn chariots. It's just they weren't in the same geographic location, to make that work out. But there was no time barrier there, and so to me, just it's so close, and that makes them, that makes us part of their story and them part of our story too. And it also makes them, these quaternary ecosystems, really relevant to all of the global change problems
Mm.
that we're facing now. And that to me is, all the cool parts aside, like, that's really powerful and important. So knowing that,
Mm.
understanding how a woolly mammoth can help us prepare, the Arctic for climate change, or to help us understand the consequences of losing African elephants, that's really powerful information to me, especially as someone who grew up in the sort of Captain Planet generation, right, where
Mm.
I've been hearing about the extinction crisis and climate change since I was a kid. And these are tools that we have to help us do some, ecological triage. We cannot, it's overwhelming if you imagine thinking about every single possible species on the planet being threatened by global change. But if the fossil record can give us some clues about where to focus our efforts, then suddenly the problem starts to feel much more tractable. And that to
Mm.
me is incredibly reassuring as somebody who cares about the planet.
Absolutely. Um, I was just thinking, as I mean, I generally am a Mesozoic paleo person,
Hmm.
but as a snow loving stereotypical Canadian, I
Yeah,
definitely understand what you are saying about kind of feeling at home in the Pleistocene. I can, yeah, I mean, looking at artwork from Pleistocene, um, like, uh, ecosystem work, I love it too, so I can completely understand you on that side.
nice. Yeah. Oh, that's awesome. I mean, we do have a
But
project that we're just starting that looks at the post KPG fern spike too. So I'll get to kind of dabble in some deeper time work soon. Yeah, no, it's all, I think, I don't know, the Earth's timeline and Earth's history is, I think, just like the greatest. It's our great story, right? It's a great story
Hmm.
of how we came to be. So
Yeah. So you had kind of mentioned a little bit before about doing a bit of field work. With your studies, like looking at the kind of Pleistocene paleoecology studies, are you typically doing field work, or do you do some like museum kind of collection work as well? And how did these two kind of interact?
we primarily do, well, our samples primarily come from the field. There is some work that's done using either data in publicly curated repositories like the Neotoma database, or there's some work that we have done using some museum collections, but primarily what we do is we go out and we collect new sedimentary archives, or we go and collect coprolites from the field. And then of course the field work is a very small component.
Hmm.
Usually we know where we're going in advance. We take a record from that place. It takes just a few days to get a sediment core or a peat core. And then we bring it back to the lab, and then it's years, years to uncover the story that's in that sediment record. With our work with the La Brea tar pits, they're doing some new excavations there, or they have been for the last few years, and we've been, leveraging those new excavations, which the previous work there, for a century had focused mostly on these large bones that were coming out of the asphaltic deposits. And the new methods that they've been using, they've been screening for the smaller components too. So
Hmm.
now we're getting plant fragments and small mammal bones, and that's allowed us to ask some interesting questions. But even though it's a museum, it's an active fossil dig, and so we're actually not working with their older collections so much. We're mostly working with the new ones. So most of what we've done has actually involved going out and taking new records, mostly from cores.
I was really lucky. A couple of years ago, I got to go to La Brea, and because I was with somebody who knew somebody who worked there, we got to go in and see some of the digs that they're currently doing, which is pretty
Oh,
cool.
yeah, it's amazing, and almost all of the digging that's done there and the sorting is by volunteers, which
Mm,
I often hear from people who are they're just so bummed that they didn't go down this path and become a paleo researcher. And I tell them, call up your local museum, because
hmm.
there's so many opportunities out there for people to volunteer. And also, I part of this work was this work was inspired by my trip behind the scenes at La Brea, and to see all the collections there. And that's when I really first started to appreciate just how much what a museum has, is
Oh, yeah.
that they put on display. It's just a tiny fraction. I had no idea. And I was having some huge museum envy. And I think if I had had that experience at a younger age, I might have ended up going down a different path and doing more
Mm,
collections-based research, because, man, it's cool.
yeah. So, other than La Brea, where else would you go kind of around the world to get these field samples from?
So we have ongoing field projects right now across both sides of the former Bering Land Bridge, so Beringia.
Hmm.
We're doing work in eastern Siberia and in Alaska. We've had a project for several years down in the Falkland Islands, which is a really amazing place to work. I love working there. I had a student who, we're finishing up a project where he did a thesis in Jamaica, which was
Mm.
definitely a change for me. And we've got some projects that I've worked. My graduate work was all done in the Midwestern U.S., and now we're kicking off some really cool projects here in New England in the U.S., both on alpine plant communities in some of our mountaintops, and also on the long history of, and we're collaborating with local tribal communities to reconstruct the long history of human land use and fire and other impacts, and sustainability here in New England.
Yeah, that sounds really cool. It
Yeah.
sounds like a big variety of things, because I can see that what you'd look at in Siberia would be pretty different from Jamaica, which would be pretty different from Midwestern US.
Yeah,
Um,
it's interesting because, in a lot of these places, like Siberia and the Midwestern US, are actually closer than you might think in terms of, well, one thing I can say is that the tools we use, the proxies, the methods, are pretty much the same in all of these places.
mhm,
Fortunately, charcoal is charcoal, right? It doesn't matter where you look at it. Um, but the pollen is another story. Some things like grass pollen grains will look like grass pollen grains anywhere in the world, and they're very helpful and friendly in that way. But when you start to look at other kinds of plant families, things get a little bit hairy pretty quickly. But like I had said, Siberia and the upper Midwest are pretty similar, and that's because most of my focus had been on the last deglaciation. So when those were
mhm,
actually tundra communities, and so tundra, boreal forest, today they're oak savannas and cornfields, but in the past
mhm,
they would have looked like very similar to large portions of Siberia. So there's actually a lot more similarity in the plant communities there than you would think. And
mm,
the Falklands, use they have a lot of the same plant families that we have, things in the blueberry family, grasses, that sort of thing. So that was actually less of a barrier, although we would occasionally get really fun things like nothofagus, these these trees that are their pollen is blowing in from South America because there are no trees in
Mm.
or at least not naturally. Jamaica was a totally different story where there's just like going towards the tropics. I don't know how my colleagues who work in the tropics do it because there are just so many more families. And pollen grains are rarely identifiable to species. Usually it's genus or family. There are lots of groups that will spread all across the world within one plant genus or one plant family in some cases. So but we had to rely a lot on reference materials and local experts, and it was a challenge for sure.
You've mentioned a lot about pollen grains and looking at the different types of pollen. I know that with paleoecology, it makes sense that you're looking at plants for quite a lot of your data. But do you ever use sort of what I would think of to me as like a typical vertebrate sample? Are you ever looking for bones and other? You mentioned coprolites, so I guess that's kind of similar. But just
Yeah.
wondering about other animal samples.
So mostly what we've worked with in the past has been, pollen and some of these other, plant macrofossils. We focused a lot on plants, but we're very interested in what animals do to plants and vice versa. And so,
Mm.
I've primarily worked with these dung fungi in sediment records as an indicator for the presence or absence of animals. So you have lots of big herbivores; they're making lots of poop, and then that is a great habitat for these fungi. And then the fungi spores end up in the lake sediments along with the pollen. And
Huh.
so the nice thing there is that you have a very good local record. Like I had said earlier, that cause and effect is super hard to untangle in the paleo. So comparing a pollen core, which is, continuous, right? So you could sample it as finely as you wanted to over thousands of years, and that record also incorporates a region of tens to hundreds or even thousands of square kilometers. You have a record of plant communities from a region, continuously through time in one location,
Mm.
and you can stitch those together and come up with some large scale maps of how communities are changing or how forest types are turning over and shifting. But then, let's say you've got a lake core from, Silver Lake in Ohio, which is one of my PhD sites. Um, okay, go find a bone near that, right? And it's maybe just one element, so one part of one organism. If you're lucky, maybe you get more, but you still only get one individual. You very rarely get lots of individuals from different, from within that community. And so you can try to stitch up what you have in the pollen record to these vertebrate databases. Like I said, Neotoma,
Mhm.
you have a record of a bunch of other fossils people have found, but they might be from quite far away. And they're going to be kind of spaced all over the place in time as well. And so you don't have that same continuity of that record. So comparing these two types of records has been super hard, um,
Mm.
which is why the dung fungus was such a great proxy, because it tells you, okay, right here on the ground with these same plants at the same time, we have, lots of herbivores or very few.
Yeah.
Um, the limitation there, though, is we don't know who's making the signal, who's doing the pooping, right?
Sure.
Um, and so, we haven't worked very much with vertebrate remains. Our work is informed very much by what those people are doing, right? So the people doing that work of synthesizing vertebrate remains is very important to help us, contextualize what's going on. But because we're so interested in direct interactions on the ground, we've been kind of limited in our ability to do those kinds of reconstructions. Um, but we're moving now towards using more ancient DNA in our lab,
Mm.
which will allow us to hopefully have more of a direct signal, a local signal of, what's actually influencing these plants on the ground, where we're reconstructing them. And the coprolites, the dung, that lets us go beyond just co-occurrence. So you have,
Mhm.
we've got this animal, we know we have this plant, but we don't know that they actually interacted in a meaningful way. But the
Sure.
coprolites can say, okay, well, yeah, we can actually tell you this animal was eating this plant specifically, and that sort of strengthens our inferences about whether those two species were interacting. So that's kind of the new directions that our lab is taking. Is ancient DNA is kind of opening up some new avenues to allow us to get at these interactions in a much better way.
That's really cool. That's kind of led me to two other questions. Now I'm trying to figure out which one I should ask first. I guess first question, because I think it's a little bit shorter maybe, is in terms of numbers, obviously it makes sense what you were saying about the vertebrates. You find one bone and that doesn't really tell you a whole lot other than that animal was there. But what about invertebrates?
Mm.
Sometimes there's, more of those than you would see with a vertebrate. But do you do any work with, any shellfish or not fish?
Um,
I'm screwing up all my words now.
oh no, it's all good. Yeah, so we and I always feel bad about this because I feel like the invertebrate people, at least in the more recent time scales, don't get as much love. And so there's very few records, or there I guess so. Most of what we can do with the invertebrate record in these lake cores or other sediment cores is to reconstruct aquatic communities, which are, I think we're appreciating much more than we used to how connected the aquatic and terrestrial ecosystems are. We, previously thought of them as much more closed systems, and I think paleoecologists have been a little bit lagging behind in terms of our understanding of the connectivity of terrestrial and aquatic ecosystems too. There was we often use this as an example. We often use this tool called loss on ignition, which is where so it's kind of a weird proxy name, but we basically burn chunks of mud at different temperatures to figure out what fraction of the mud was organic versus inorganic versus carbonates. And that can the organic component is thought to tell you something about how productive that lake is, how productive the community is. And that kind of correlates with temperature. So warmer tends to be more productive. And everyone I ever talked to or learned from, would say this is, really telling you something about the aquatic community. You can't interpret this as a terrestrial signal. It's not, picking up the anything going on with, what's happening on land. And then I read a paper by a colleague of mine, Andrew Tenensap, where they actually traced where the carbon came from in some of the aquatic ecosystems that they studied. And they found out that these like fishes in this lake, and I think it was in Quebec, were something like 65% maple tree. Basically, like,
interesting.
The maple leaves would fall into the lake, and the invertebrates would eat those, and then the fish would eat the invertebrates. And so if you trace it back, like the carbon source is really these fish are basically made of maple leaves, which just I think reinforces this idea that there are more connections among these terrestrial and aquatic communities than we thought before. And so that's a very long-winded way of saying that here we are back again at big picture scales. I think is really cool. B, I think we should always be challenging ourselves in terms of what we think the limits are and what we can understand with these proxies. And C, in paleoecology, and at least in the Quaternary, these lake sediment reconstructions where we look at invertebrates, oftentimes we just treat them the same way that we used to treat pollen, which is these are a paleoclimate proxy, right? So a lot of these organisms like chironomids, which are these non-biting midges, they're little, when they're in the larval stages, they'll leave their little head capsules behind. And oftentimes, people just use that as an indicator of past climates. But, we can reconstruct multiple components of these aquatic food webs, including, things like diatoms and other proxies, even fishes, whether that's with, bones or now with ancient DNA that's in the sediments. And we can actually ask, questions about the community ecology of these ecosystems too. We have not done that very much yet in my lab. We have, for the most part, kind of treated these invertebrates as regional paleoclimate indicators. But I think that there are a lot of cool frontiers where we can do a better job of just treating these aquatic ecosystems as fun communities to understand in their own right, and not just leaning on them in terms of what they can tell us about the terrestrial record.
Yeah, no, that makes sense. It's quite interesting. I hadn't really thought about that with kind of invertebrates. I'm used to the typical paleoclimate, it tells you about the temperature and that kind of thing. So it's an interesting thought about what else you can do with them.
Yeah, and I think part of that is when I started this job at UMaine, I was hired at the same time as an aquatic ecosystem ecologist who looks at community ecology, and actually studies terrestrial and aquatic linkages too. And just talking with him has gotten me thinking so much about, wow, okay, the same things that you look at in your, he works with, these little mesocosm experiments where they'll make artificial ponds or he'll sample actual lakes or ponds. Um, it's like you're using the same tools that we use, but you're looking at them in the modern record and trying to understand, competition and dispersal and other interesting questions. But
Hmm.
we actually have long-term, sedimentary records of the same species that you're examining. But we tend to, like I said, we tend to not as think of them as often as community, members.
Hmm.
And the same thing was true. A lot of the early applications of the pollen record were for reconstructing past temperatures or past climates. Um, and but then people started to appreciate and look at these forest communities as actual sources of interesting community data for their own right. So I just think for whatever reason we've kind of lagged behind a little bit in our understanding of paleo aquatic communities, at least in the more recent quaternary.
Yeah, and then I guess the other question that I was wondering about was you were talking about the different kinds of dung and how you can tell if there's a herbivore. Um, is there anything else that you can find out other than when you look at the coprolites? Is there anything other than just this was a herbivore or this was a carnivore that tells you what these animals were?
So we're hoping to use ancient DNA to actually figure out who made the dung, who the pooper was, so to speak,
Yeah.
and the I guess the depositor. And but we've also been doing scanning, like high resolution scanning of the physical dung samples themselves before we cut into them and sample them, because we're hoping that shape and morphology of the dung
Ah.
might be able to tell us something about who created it. Because you can identify a lot of modern animals based on the just physical characteristics of their poop. So,
Hmm.
imagine, deer versus rabbit versus something else, skunk or whatever.
Hmm.
So we're hoping that that will pan out. We have to have a good enough sample size and enough representation of different animals, and then we need this external corroboration, right? We didn't see the animal lay the dung down on the landscape, so we need this,
Hmm.
this sort of third party validation, which we're hoping that the DNA will give us. And then within that dung, there's a ton of different proxies that we can use. Pollen, plant fragments, DNA. We'll be looking at diatoms to see if we can characterize something about the water quality. Are they what kind of water are they drinking? Are they eating, or are they? If we have snow specific diatoms, that might tell us they were eating
Hmm.
snow. What season they died, right? So there could be some information there. We'll try to look at some other chemical indicators, but yeah, there's a lot that we're hoping to be able to pull from these coprolites.
So many things I didn't know that you could do with poop.
Yeah, yeah, yeah. The treasure trove of information.
Yeah. I guess, we've been talking about the Pleistocene, and obviously, one of the big questions that people always talk about is the big megafaunal extinction. Um, can you talk a little bit about that and some of the work that you've been done looking at exactly what happened there?
Yeah. So my interest in these extinctions actually, began when I was first applying to graduate school. I had been, interested in these big animals from the beginning. They were kind of, like I said, my hook into the Pleistocene. And I had originally approached the person who became my graduate advisor, Jack Williams, at the University of Wisconsin, because he had done some work on these late glacial novel ecosystems or no analog communities. So these are forests that are compositionally unlike anything we have today, even though all the species are still around, they're just growing in different assemblages. And he had mentioned in a paper that actually was from his dissertation that, we might not know about the role that large herbivores played, because it's really difficult to study. And at the same time, I had been learning about this dung fungus that I had mentioned that allows you to test questions about the presence and absence of herbivores. And so that was
Hmm.
sort of my pitch to him. And that was kind of the first, that ended up being my master's and PhD research, was using these tools to test the role that large herbivores may have played in the formation of these ecosystems. When I started that work, I thought that because these were no analog, communities with, we don't have big herbivores today. I thought that these no analog forests, these forests without a modern analog, they would have been around because the herbivores were there, right?
Hmm.
So the herbivores were creating them because, mammoths and horses and camels and other things we had roaming around North America were creating unusual, ecosystems. What actually happened was, as soon as so if we looked at the dung fungus, as soon as the dung fungus dropped out of the sediment record, within decades after that, that's when we saw the formation of these forests. And that really
Yeah.
surprised me. And I was like, that's not what I expected at all. And that's when I started thinking, okay, well, maybe these extinctions have consequences for these plant communities. And we had been, kind of collectively as a field, thinking about the relationship between megafauna and plants as, okay, the megafauna are, these herbivores are relying on these plant communities. That's their habitat, that's their food source. And so it's the plants that change, and then that's what then drives these declines in these large herbivores. But what my records were showing was the order of events was different. It didn't support that conclusion. And so
Mm.
I started thinking about vegetation change as a consequence rather than a cause of these extinctions. And that just opened up so many avenues for me in terms of my interests in biotic interactions, in the interactions across trophic levels between plants and animals, which can play out in so many different ways. And so we still research that in other systems, including, mega herbivore and vegetation communities. But that that kind of started with that surprise that I wasn't expecting that result.
Mm.
And so that started, getting me thinking about, we have a lot of debates about the relative role of humans versus climate change in driving these extinctions. I think it's really difficult to explain these extinctions in the context of many cycles of glacial interglacial, periods, unless the one thing that's different about our current interglacial is the spread of humans across the globe. And so I think climate change is important. It, definitely impacts populations. It probably, extinction is very often a sort of 1-2 punch. It's not usually single cause. But, it's hard for me to imagine, that if humans had not migrated out of Africa, that we wouldn't, still have these big animals roaming around today. Um,
Mm.
and but on the other hand, I'm often less interested in the causes of those extinctions, which is what everybody wants to focus on. um,
Mm.
but for me, because I'm so interested in conservation messages, we know why big animals are going extinct now, right?
Mm.
There's no mystery there, and so and functionally, it may not matter in the end, which was more important, humans or climate change, in causing some of these extinctions in the past.
Mm.
Um, but what I felt was missing from the story was, what happens next, right? What are
Yeah.
the consequences? What do we lose when we lose a woolly mammoth? What's lost from the landscape? And so for me, I'm often much more driven by a desire to understand what these animals are doing to the landscape than
Mhm.
why we lost them.
Yeah, that's a really good point that, everybody always, why did they go extinct? That's what everybody always thinks about. But I guess thinking about the consequences of animals moving in or moving out of areas kind of leads to some of your new work and this paper that is about to come out in the Falkland Islands. So, in this work, you talk about the effect of seabirds and the role that they played moving into the island ecosystem. So, can you talk a little bit about that work and what role seabirds play, kind of moving away from megafauna?
Yeah, that is such a fun story. I had spent my graduate work working with on the impact of large herbivores and their impact on trees,
Mm.
and then I shifted to an island with no native trees and no native land mammals. So like the dominant herbivore in the Falklands is a goose, right? And so
Mm.
it's a really cool place, a really interesting place to be, and to work. And so I was initially drawn to the Falklands while I was finishing up my postdoc. I had gotten my job at UMaine and I hadn't started it yet. And the South Atlantic Environmental Research Institute, which is based in the Falklands, was reaching out and developing partnerships for people to do environmental research in the Falklands, both as part of their interests in protecting biodiversity in UK overseas territories, but also for economic development in the Falklands and just lots of really good reasons. And so my first thought was, well, I don't really know much about them. So I Googled, I Googled the Falklands and I read, because we got this email saying, does anyone have any project pitches? We're going to have a meeting and you can each give a couple slides about things you'd like to do. And then I read that, a large foundation of the Falklands economy is there's ecotourism, fishing, and sheep. And that there had been a lot of, about over a century of overgrazing.
Mm.
The native tussock grass communities in the Falklands, which are really important for seabird habitat, had been, grazed down. And these grasses, which were once widespread across the islands, at least in the coastal areas, were now, decimated, literally decimated, or more.
Mm.
And there had never really been a vegetation assessment. And I thought, well, okay, that's what paleo can do, right? We can provide some information about this.
Mhm.
And when we went down and visited, so we were encouraged to come and visit, and they liked our project idea. And when we went and visited, we saw that they had been trying to do some tussock planting, these grasses that are, can grow meters tall. They're it's so interesting. It's like you're sitting there and you're surrounded by this lush grass, and there's penguins, running up and down these, white sandy beaches with like this turquoise water, and you think you might be in the tropics, but then there's penguins everywhere, and so it's a very, very like fascinating place to be. But Yeah, so we were there, and we saw that there had been these attempts to plant the tussock, and it's been really mixed, and it's often hard to get it to establish. And we had talked to a farmer who had said, who was not living on the coast, which is where it's conventionally thought that, this grass can only survive by the ocean. It can't
Mm.
thrive in the inland. So if you go to the inland part of the Falklands, it's just this brown landscape.
Mm.
It's this grass called whitegrass. It never greens up. It's brownish all the time. And this farmer told us, this sheep rancher said, well, I don't understand what the problem is. I grow it all the time. My chickens use it as a windbreak. And that was like the moment of like a light bulb, like, okay, chickens, poop. All right, nutrients. Like maybe
Hmm.
that's what's going on here. And if you fence out the sheep to get, so that they can't get into the grass, maybe the seabirds can't get in, and these grasses might need this nutrition from seabird guano or from chicken poop or whatever.
Mhm.
Um, and so then we started taking peat cores and reconstructing we use these geochemical indicators from seabird guano. So heavy metals, things like copper, arsenic, phosphorus, other things, to tell us when the seabirds were present or absent. So we had this indirect indicator of seabirds because of their guano chemicals.
Hmm.
We reconstructed nitrogen from which is like the basically the fertilizer from their guano. And I should say here, when I say we, I didn't do any of this. This was my PhD student Dulcinea Groff. She did all of the work. This was part of her dissertation. She is now a postdoc at the University of Wyoming. She's the lead author on this paper. Um, and she also looked at the pollen in the record to establish when these grasslands became established. She looked at
Mhm.
charcoal to figure out when fire, or if fire was a natural part of the ecosystem, because the land managers in the Falklands have had some debates about whether burning is natural or not, whether they should burn to promote better forage for the sheep. I mean, there's so many questions about land management there. And so what we found, which is really cool, is that around 5,000 years ago, so we have a 14,000 year record from a place called Surf Bay, which is close to Stanley in the Falklands, the main little town of about 3,000 people. And so we have a 14,000 year record. We have no indication of seabirds being there until 5,000 years ago. And all of a sudden, seabirds show up. And within, a century or so, that's after that, that's when we see the establishment of these tussock grassland communities. So we see a big increase in peat productivity, because these grasses, produce these really rich peat deposits. We see a big increase in grass. We actually see the first fire showing up. So we think that there might be a role, like fire in these systems are probably natural, but they're probably fuel limited, which is true for grasslands. Grasses tend to be fuel limited than ignition limited. Just need enough material to burn. And so basically, if in the chicken or egg story of which came first, the tussock habitat or the seabirds, it seems to be that the seabirds show up first. And there's a couple things that are interesting about this, and one of them is we don't know where they came from, right? So did they come from another location in the Falklands? Did they come from another island? Did they come from South America, Tierra del Fuego, Antarctica, some other location? And that's a little bit alarming because in the South Atlantic 5,000 years ago, when these animals were establishing, this was a period of cooler climates. So there was a big drop in regional temperature, sea ice expanded in Antarctica. And so that suggests that the Falklands are a colder climate refuge for these seabirds. And that's the opposite of the direction that we're headed now. And if we don't know where these birds are going to go when the climate gets warmer, that's a little concerning because those places that those seabirds might be headed to aren't necessarily protected today in the same way that, their current habitats are. So we should be expecting, there could be pretty abrupt changes where these seabirds could just pick up and move somewhere else or try to if those places are available. So that's the first thing. We need more information. We need more of these records to tell us where these birds are going. But also it tells us too that, if we want to restore some of these coastal habitats, we need the ecosystem services that these seabirds provide. They're also engineers kind of creating their own habitats in these coastal systems. And, these low, sort of lower productivity island grasslands need the nutrients that, the oceanic nutrients that these seabirds are providing. And so I think that, that's something that we can hand to conservation and land managers in the Falklands right now. And we can say, either get some seabird poop or some close equivalent or some chickens or something. And that might help you, reestablish these tussock grass communities. The other thing we don't know is which birds are making the signal, right? So it's the same problem we've had with the dung fungus, right? Where we know that it means that there's animals here, big animals, but we don't know which species specifically are kind of creating, creating that dung signal that the fungus is responding to. And here in the Falklands, there's some indication from some slight changes in the chemistry of the sediment that we might be seeing some shifts from maybe one or two species to another. So we would love to try to figure out more about, are we talking about, there's multiple species of penguins, there's shearwaters and petrels and a bunch of other birds. Which ones are we talking about here? Not all of them have a relationship with tussock grass, but a lot of them do. Some of them burrow into the peat under the grass. Some of them nest on top of these peat pedestals. So they all have kind of different relationships. Sea lions also come into the tussock grass to take refuge from the wind. They'll actually sleep on top of these tussock grass, pedestals, because the peat decomposes and it's warm. And so they'll just climb up and flop down on top of them. Oh my gosh, there's one scary moment in the field. You asked about field work, where we had been at this one site doing some modern vegetation surveys, and there were all of these male sea lions all hanging out down this cliff, kind of in this rocky pool, and we could hear them all the time down there, kind of like roughhousing and making a lot of noise. And then one night, I just woke up in the middle of the night and I could hear them all around me. And they had come up off of the cliff, up onto the area where we were, and they were just like making their way into the tussock and like flopping onto these tussock pedestals. And sea lions are big and scary.
Yeah.
And usually what they'll do is they'll just chase you, or if you get in the way, they might take a bite. But all I could think of in that moment was, please don't flop on top of my tent. Like, please do not think that like my nice warm tent is a good spot for you to sleep. I do not want it under hundreds of pounds of sea lion. Yeah, so they're up
No.
in there too, and they're pooping and doing stuff as well. But yeah, it's a really cool story about how these connectivities among these ecosystems,
Yeah.
like that to me is where I just think that's really cool. So
Yeah.
similar kinds of questions, similar kinds of tools, totally different part of the world from where we had worked
Yeah.
before, and we're still seeing again the important like take home messages are everything's connected. You can't just study these things in isolation if you want to understand these sort of broader patterns.
Yeah. I guess, I mean, to be honest, that seems like a pretty good way to finish up talking about this paper. I was going to ask just one question, so I'm not going to end on that.
Sure.
Although it was a good end, but I do have one question,
Yeah.
which is just, you had mentioned before about like the ancient DNA and looking at the coprolites, but I'm guessing that that's a little bit more difficult to do in this case, since you mentioned that what you were looking at to find the seabird record was more kind of the heavy metals that you find. So I suppose you're not finding, an actual definite deposit or something.
No,
Is there?
yeah,
Yeah.
it's a lot more liquidy, and it just more
Yeah.
cohesive. But so we thought about DNA, but the problem is these peats are super acidic, which is great for preserving lots of things, really bad for DNA.
Yeah.
And so even just finding bones is really hard. Even if you go into some of these penguin colonies or other seabird colonies, finding bones can be really tricky. So what we're thinking potentially of doing, I guess there's a couple ways forward. You could try to take some DNA samples from modern birds and try to look
Mhm.
and see if there's any genetic fingerprints of past colonization or isolation. You can often read these things in the genetic code of the organism, these kinds of dispersal events or isolation events. So that might be one way forward. We'd have to collaborate with somebody because that's not what we do. But the direction we're leaning is, okay, well, maybe different, if these different birds are eating different things, then maybe the community ecology, if you will, of their heavy metals, right, are going to there's the assemblages of their heavy metals in their guano might be distinctive or species specific. And so
Mm.
what we want to do is go back and get a whole bunch of different dung sample or guano samples from a bunch of different species of seabirds and try to characterize the abundances of different heavy metals to see if we
Mhm.
can come up with a match for, okay, well, when we see, these elements are high and those are low, maybe it's this bird. And when things shift a little bit, maybe
Interesting.
it's this other bird. I don't know if it'll work, but I hope that it does. Because and if anyone else has an idea of how to potentially do this, where we don't have good preservation of things like DNA or soft tissues or bones, I would love to hear it. But yeah, right now, or maybe there's some parasite that we could try to pull out of the sediment record. But yeah, sometimes we can get feathers, but not as much as you would think, considering how many
Mm.
birds are in this spot. So yeah, we're scratching our heads about this right now. But the first lead we're going to chase down is to see if there's some chemical fingerprint of individual bird species that we could then somehow pull out of our very generalized record.
Mm. Well, fingers crossed that you can figure that out because it sounds really cool.
Thank you. Yeah, and it's really funny. We keep coming. I do not know how this happened, but somehow poop has become this unifying theme of all of the different projects that we've worked in, and it's, I mean, it's a super useful tool. It was not intentional. I do not have an obsession with species. It just, yeah, it's just right. It's just everywhere, and it's full of stuff, and it's useful.
I've certainly learned how useful it is today.
I'm glad.
Well, I guess maybe just kind of to wrap up, can you say a little bit about like the next big thing that you're working on and maybe what you think is another big question that needs to be looked at moving forward?
Oh, that's a good question. Let me think about that in terms of something we haven't mentioned yet. Um,
Or even just,
I think
what's next on your radar.
I think one of the things that's just getting off the ground now and that we're really excited about is our partnership with local Indigenous communities. Um,
Mm,
and I think, and I know we've mostly talked about plant and animal interactions, but of course, most of these places that we've worked have had people for a really long
mhm,
time. People have been here in Maine since the ice retreated, right? And as paleoecologists, as quaternarists, we have historically been pretty bad about collaborating with and Indigenous people and valuing Native knowledge.
mm,
And the people who were here are still here.
mhm,
We often talk about them in the past tense, but they're still present today in Maine. They have knowledge, they have oral histories and traditions about their own, land use. So
mhm,
I'm really excited to make those partnerships, both in terms of how we can learn from them about their relationship with the land through time, but also how we can provide information that they might need for questions of tribal sovereignty.
mm,
In my conversations with local tribal communities, we're learning that, for example, a lot of some of their most basic needs are just getting their ancestors' remains, repatriated from museums, right? And so
mm,
providing information that backs up their oral knowledge of where they've been can be
mm,
super useful, just for being able to, because the burden of proof is on them to say, yes, those remains belong to our people because we've been here through time. And that really blew my mind, just in terms of, just how we can potentially provide information that can help them with the questions of their own sovereignty that are important. So I would say, I think, I'm seeing more and more of this appreciation for collaborating with and co-producing knowledge with Indigenous peoples, and I think quaternary paleoecologists in particular can do a much better job of this. And I'm
mm.
hopeful that that's a great new direction for the future for us, because we can't just
Yeah, I
keep.
think.
Yeah, we can't we work in these landscapes that have been inhabited. We often talk about human impacts, or if it wasn't climate, maybe it was people causing these fires, but those people
Mm,
have names and they have cultures and they're here and we can talk to them and build trust, relationships on trust. And that's going to take a lot of work, a lot of work. But I think it's really important, and I'm really hopeful that this is a new direction for our community.
yeah, I think it sounds really interesting and definitely like you can probably learn quite a lot from actually just talking to them and finding out, what their history was of the area. Because really, we should know that, but we've never bothered to ask. So,
Yeah, exactly. Yep.
oh, that was great. Thank you very much, Jacqueline. I really enjoyed that.
Yeah, me too. Thanks so much for chatting today. It was really fun to talk to you.
Paleocast was brought to you by Dave Marshall and Liz Martin Silverstone, with contributions from Tom Fletcher, Vish Venkat, and Elsa Pancharoli. Music was composed by Patrick Kendall Smith. Paleocast was made possible by funding from the Paleontological Society and the Paleontological Association, but the show now relies on funding from you, the listeners. So, if you've liked this episode, please consider donating, and thanks to everyone that's helped out so far. Please visit paleocast.com for the supplementary material to this episode and for our archive of past programs, and follow us across social media platforms to get all the latest news. Finally, if you enjoy our podcast, then please explore all of our video content on YouTube and follow our other projects, the Virtual Natural History Museum and the Paleocast Gaming Network.